Rotifers are microscopic freshwater-dwelling multicellular organisms. They're already known to withstand freezing, even in liquid nitrogen, boiling, desiccation, and radiation, and the group has persisted for millions of years without having sex. The humble yet remarkably hardy bdelloid rotifer has now surprised researchers yet again. A recent study unearthed 24,000 years old Siberian permafrost and found living or at least revivable rotifers there. Surviving 24,000 years in a deep freeze is a new record for the species. Rotifers aren't the only living organisms to emerge from permafrost or ice. The same researchers behind this latest discovery had previously found roughly 40,000-year-old viable roundworms in the region's permafrost. Ancient moss, seeds, viruses, and bacteria have all shown impressive longevity on ice, prompting legitimate concern about whether any potentially harmful pathogens may also be released as glaciers and permafrost melt. Given that bdelloids are generally only a threat to bacteria, algae, and detritus, however, there's not much need for concern regarding this particular discovery. But as key players at the bottom of the food chain, newly re-emerged rotifers indicate that maybe we should think about how species that haven't been seen for millennia might reintegrate into modern ecosystems. The Soil Cryology Lab in Pshchino, Russia, has been digging up Siberian permafrost in search of ancient organisms for roughly a decade. The group estimates the age of the organisms it finds by radiocarbon dating the surrounding soil samples. For example, last year, the researchers reported a frozen zoo of 35 viable protists, which is a nucleus containing organisms that are neither animal, plant, nor fungus that they calculated ranged from hundreds to tens of thousands of years old. In their most recent discovery, the cryology researchers found the living bdelloids after culturing the soil samples for about 1 month. Among rotifer classes, bdelloids have the fairly unusual ability to reproduce parthenogenetically by cloning, and so the original specimens have already begun to do so. Although the clones made identifying the ancient parent challenging, this did greatly facilitate further investigation of the characteristics and the behavior of the unfrozen strain. Throughout all of the above permafrost studies, there is always the concern of sample contamination by modern-day organisms. Besides using techniques designed to prevent this, the team also addressed this issue by looking at the DNA present in the soil samples, confirming that contamination was highly unlikely. The phylogenetic analysis furthermore showed that the species didn't match any known modern rotifers, although there is a closely related species found in Belgium. The team was naturally interested in better understanding the freezing process and gaining insight into just how these rotifers survived for so long. As a first step, the researchers subsequently froze a selection of the cloned rotifers at -15 degrees centigrade for 1 week and captured videos of the rotifers reviving. The researchers found that not all of the clones survived. Surprisingly, the clones generally weren't much more freeze-tolerant than contemporary rotifers from Iceland, Alaska, Europe, North America, and even the Asian and African tropics. They were a little more freeze-tolerant than their closest genetic relative, but the difference was marginal. The researchers did find that the rotifers could survive a relatively slow freezing process. This is noteworthy because it was gradual enough that ice crystals formed inside of the animal's cells, a development that is usually catastrophic for living organisms. In fact, protective mechanisms against this are highly sought after by anyone in the business of cryopreservation, making this latest finding especially enticing from that perspective. Although the authors aren't quite in that business, they do plan additional experiments to better understand cryptobiosis, the state of almost completely arrested metabolism that made the rotifer's survival possible. As for research into cryopreservation of larger organisms, the authors suggest that this becomes trickier as the organism in question becomes more complex. That said, rotifers are among the most complicated cryopreserved species so far, complete with organs such as a brain and a gut. Which returns us to the questions of what other organisms might reappear with a warming climate, and what impacts they might have. Evidence so far shows that multiple types of organisms are still alive in the ice. At least on a microscopic level, it seems possible that intact microecosystems may thaw together. How these long-dormant species will compete or coexist with modern ecosystems is difficult to predict at this point, but it's probably worth further consideration.